And then Miguel, as it turns out, actually could join us today. So we're utterly delighted that we can welcome them both. So what I'll do is first I'll introduce them a little bit more appropriately, and then talk a little bit about the book, drawing your attention to some of the features of it that I think are particularly useful and very, very important for Egenis' work, broadly conceived. Then I'm going to give the word, of course, to Miguel and to Jim, who will give their own perspectives of the book and also on their current work, kind of linking it up and giving us a sense of where this is all going. And then we'll open it up in the last half hour or so for discussion. So this is how today is going to work. So let me just then introduce the two speakers, first of all, and the two authors. So Miguel is now a senior lecturer in Edinburgh, and together with our colleagues in the Department of Science, Technology and Innovation Studies, and he's now been there for about maybe
10 years.
No, 10 years.
Oh God.
Yeah, this is the pandemic. Always at 3 years. Whatever you think the number is going to be, 10 years. And indeed, he's been there pretty much since he published his last book before this one, which was called Biology, Computing, and the History of Molecular Sequencing from Proteins to DNA, that came out in 2012, and it did very much kind of lays the groundwork for then what has become a lot of the work that's featured in this book. And one peculiarity of Miguel is that before getting into academia, he worked as a journalist. And so he retained excellent interviewing skills, but also this flair for being able to pick up things that are particularly relevant, and then also being able to talk about it on a variety of platforms. He did his PhD at Imperial College London, and we were just reminiscing during that time. We came here a fair amount of time to kind of pick up on what work was happening with Egenis then. And he then worked in Manchester at the Center for Historicise. He worked in the Spanish National Research Council in Madrid, where I was very pleased to come and see you, I remember at that point. And then he's been in Edinburgh indeed for the rest of the time. And yeah, and of course, we'll hear much more about Miguel's research in a moment, but certainly he's been one of the people that now is absolutely internationally recognized to be one of the key contributors to thinking about the history of sequencing and genomics, and particularly the history of genomics beyond the human, which is something that, of course, in Egenis we adore as a topic and informs a lot of the work that's going on here too. So we're very grateful for that. Thank you, Miguel. And Miguel was also the principal investigator of an ERC grant where he managed to employ Jim. Now, Jim Lowe is somebody that many of us know very well because he did his PhD here with us in Egenis around a number of years with me and Miguel. And since then, he worked for a while with me and Gail Davis on a project on basically pharmacology broadly conceived, and then since he's been off to Edinburgh and joined the company of our wonderful colleagues there and working in Miguel's group. And of course, you see the wonderful results of that collaboration here, and yeah, I think it's just a wonderful thing for us also always being to think, oh, how great that one of our students has ended up doing such wonderful work in Edinburgh. Jim is now back in Exeter, actually, for the next 2 years, right? 18 months or so. Working with our colleagues in the, you're based in the business school?
No, it's Centre for Ecology and Conservation.
Okay, sorry.
It's really new though, it's
I know, I just never know what people are. Okay,
yes.
so you're in the Centre for Ecology and Conservation, but it's part of the big renew project that we've heard a lot about in the last few seminars and this term. So we're all, I think, pretty updated on what is going on. Eleanor was just here a couple of weeks ago giving us the lowdown on where things are, which is great. So Jim is around for anybody who wants to catch up with him and discuss a little bit more this work also beyond today. Now, the book. You see it here, and maybe you want to pass it around, like so people can have a look. And we can also pass around the leaflets that give you a discount if you want to get it. The nice thing, of course, is that this book is in fact open access online, so anybody who wants to get it, also immediately those of you who are online and watch it, you can download it if you haven't done it already directly from the Palgrave website. And yeah, I mean, it's just a wonderful achievement for a number of reasons. And one of the reasons why, one of the reasons that Jim and Miguel provide in the book is that it challenges directly this hourglass narrative of how we think about the history of genomics, which is so common, especially once you go a little bit further up from HPV and history and philosophy of biology. And even I think quite a few of our colleagues in social studies or science sometimes kind of, adopt this in a way that's really infuriating, which is this idea that, There was once molecular biology with all its complexity, and then the Human Genome Project happened, and so everything got funded and absorbed by the Human Genome Project. Since then, we have this thing called post-genomics, which is like when things are complicated again, and so the hourglass gets broad again. And they do an absolutely fantastic job in this book in a variety of different ways of demolishing the historical narrative and provide a lot of the substantive historical material to really think instead about how diversified that history has been. Of course, they're not challenging per se the idea that we are looking at, in many ways, a reincarnation of big science, as many people have argued in biology at that point in time, and not a coincidence that Los Alamos was one of the big places where some of these kind of library efforts around genomic data were performed. But they're enriching it by showing just, at least many other locations, many other species of this work is done, and also a lot of the things, both technologically, conceptually, and biologically, that had to be researched and constructed so that something like the Human Genome Project would take place in the way in which it did. And I think it's incredibly important history in a huge variety of ways. And of course, perhaps very important because also they put a lot of emphasis on the materiality on some of those practices. So one of the parts I love the most in the book is the emphasis on the different types of libraries, and particularly, first the yeast artificial chromosomes, so the YACS libraries, and then the BACs, the famous BACs, the bacterial artificial chromosomes, which basically started, as they remind us, were created in 1992, the first exemplars, and have become such an important source of information and utterly, completely complementary to actual sequencing. I mean, without that, we wouldn't have absolutely anything around GBank and EMBL and all the kind of big collections and databases that we're used to thinking about. But these libraries hadn't really been documented properly. I don't think. I mean, this book does a fantastic job of really telling that story alongside the story of genome sequencing as people usually think about it. So that's just great. Thank you for doing that. The other thing that I think is really important, of course, is the fact that, as the title says, this is a history of genomics across species, and they're looking specifically at the efforts around yeast and the efforts around the big sequencing, which is a wonderful thing because, of course, the moment you're doing that, that brings you into a whole different ballpark for what sequencing even meant, how people conceptualized this, what were the goals for this, which communities were considered in interaction with this, and of course breeders come forward as a really important element here, as do brewers, which is also wonderful, both in terms of what it meant in terms of the locations that were adopted for this work, but also in terms of the kind of conversation that happened around it, which I think is also just really great. And in so doing, again, expand the view for not just, what does it mean to do genomics today, but also how it came about, that we had a certain set of conversation around it. And certainly me and Rachel Akin have done some work looking at other model organisms, especially in the 1980s, 1990s period. Quite a few people, Stephen Hilgard and others, have pointed out, look at the conversations that are happening across all the model species at a moment where the Human Genome Project is being shaped, and in fact, many of those conversations really were, the active prod and continue to happen as all through what was happening with the Human Genome Project. But this book actually goes beyond this, but because looking at something like animal breeding as one of the fuels for how we think about sequencing, I think is really pushing the envelope in a really fantastic way, which really, again, speaks to many of the things that are happening in Egenis at the moment and for many of our colleagues here. So this kind of complexification of this genomics is really, really useful. They're also very useful at the beginning of the book. I don't think we're going to be talking about this who knows later, so I'll just mention this. They actually do mention the conceptual question of what this does to the definition of genomics, which is discussed in the introduction. I recommend that you have a look if you haven't. Of course, very aware also of what the work that's been happening here, particularly John's work on thinking about that issue. And they're thinking about genomics in relation to two main properties. One is the systematicity of actually thinking about the genome. So basically saying, for this to be genomics work, it needs to be work that looks at a variety of genes in relation to each other and to the broader kind of for the broader organism, and also the treatment of the genome as the substrate of its efforts. This is the way in which they define this idea that it also has to be anchored in this question of conceptualizing the genome in a very particular way as a substrate for biological operations, which I think is absolutely fair, and is a very nice definition that then manages to embrace the various activities that they're thinking about, and also, again, move the goalpost away from this idea of comprehensiveness, which is, again, what a lot of people tend to associate with genomic efforts, and is such a completely useless way of thinking about them. And the other thing that they draw a lot of attention on, which I think is really important for us, is the processual nature, of course, of not just obviously what genomics is, but how it's captured. So, and in particular, a lot of the book, the final part of the book is devoted to the idea of reference genome, which a lot of people right now, including me, I've had many conversations about reference genomes just in the last 2 years. Many people are interested in this. And the key point that they're making is that reference genomes are themselves fairly dynamic entities that keep moving. And we know this through the history of the actual sequencing process and of projects. And this is now even more important now when people are starting to diversify this more and more. I think that gets very close to some of the things that John have been trying to argue about the kind of reification that happens when you take something as a reference, thinking that this would be functional beyond the time and the place at which the reference has been created. And there's lots one can say about this. I suppose one of the questions I had about the book is, to which extent maybe you've been fair to some of the effort that actually have been put into going towards a reference, right? So I mean, Tiger and all of that some of the efforts that were happening, particularly in the States, but also partly in Japan, partly in Europe, additionally to provide a comprehensive genome, but actually to give us the kind of quality standards that we need to create a reference. I mean, of course, because you put so much emphasis on this diversification, and I guess, but you cannot do everything in this book, right? But I was wondering, I suppose part of the other part of the story would be to say something more about the quality of those standards and the diversifying quality of the standards around the world. I mean, we know, for instance, there's been so many issues between China and the US already then around the issue of what constitutes a reference, because in the States, and Tiger basically didn't accept any of the Asian reference sequencing, whether it was Japanese, whether it was Chinese, not like we have to redo everything in American terms, right? And in fact, it even happened for some of European history. So I mean, there's so many things to be said about, which hopefully you will do in the future, about how the question about reference is played out, even in those respects, and what can we, how do we balance out the kind of political economic history of that with the actual questions around whether there were standards that would make more sense biologically than others, and why, and in respect to which technologies and which resources. Right? So there's a whole really interesting discussion that starts in this book, but I think will go well beyond that. And the last thing I want to put an emphasis on before I give the word to them is the geographical issue, which is just great. I mean, I was so happy to see Europe take a central role in the book. I mean, basically, in fact, the whole book runs on the question of the political economy of European funding and European conceptualization of decentralized research in a very big way. And that's something that many of us have been thinking about for quite a long time, but it's just wonderful to see it articulated nicely in this book, partly because you're really documenting some of the history. Of course, to document the history fully, gosh, there's not such a thing, and that would have taken a much broader project with even more countries. But the fact that, for instance, you document the German, particularly the German effort around sequencing and particular sequencing. I mean, Germany really was leading, and Brewers, anybody, really was leading on that research between the 1980s and 1990s. I mean, that's just priceless. Really important, because also it sheds light on the tensions, for instance, there were between EMBO and the EMBO organization as a European organization, and the EMBO as a UK-based organization, partly funded by the Wellcome Trust, which is really interesting to read across the lines in your history. Like, you can see some of those tensions starting to emerge already in 1980s and 1990s. Of course, now with Brexit and lots of other things that happen in the meantime, there's all sorts of issues around how these things actually relate to each other. Now one of my PhD students, Natalia Sheehan, is actually at the EBI trying to document what is going on at the moment with the nucleotide collections and how they're related to what is going on in Edinburgh more broadly. And there's all sorts of very interesting issues there around how those relations have been shaped. So to see you giving this very nuanced account, both on the European side of what's going on in Germany, in the Netherlands, and especially some parts of Western Europe, how European funders are conceptualizing the need for decentralized research, and the fact that also there was such a strong emphasis on not conceptualizing sequencing as the beginning and the end of the big effort, like going beyond the technicalities of it. I thought that was just a really, really interesting way again of pushing the envelope on some of the things that we've been doing. And of course, there are the Sanger Institute. I mean, if you're interested in that, you want to read a book that really does give you a sense of where to situate the Sanger. In fact, quite how important it really was, especially once you put it in relation to all these other efforts around the world, then this is it, because there's a couple of chapters here that are just fantastic in that respect. So I think I'll leave my comments here, just trying to pick out some of the things that may come up in discussion later on also. And thank you very much again for doing this, and give the floor to Miguel first.
First Miguel. Okay. Well, thanks a lot, Sabina, for a very kind, introduction. Thanks a lot to Egenis, for giving us the opportunity to come here and present the book and to be back here, because as Sabina said, I used to come a lot when I was doing my PhD in London. And well, as Sabina indicated, this book is the result of a project that was funded by the European Research Council and was conducted at the University of Edinburgh between 2016 and 2022. And in a nutshell, what we wanted to do was to explore the history of genomics across 3 different species: yeast, Homo sapiens, and pig. Why we chose these 3 species? Well, because they are very different types of organisms. Yeast is a fungus that has got a long tradition of use in the brewing industry, and more recently in the biotechnology industry. The domestic pig has been the subject of breeding programs within the fields of both quantitative and molecular agricultural genetics. And then Homo sapiens gave us the opportunity to look at how genomics is used in medical research and evolutionary biology. And also, quite crucially, these 3 species were the subject of concerted projects to map and sequence their genomes in time periods that partially overlap, but that were somehow continuous chronologically. So by looking at the 3 of them, we have a wide chronological landscape of genomics from the mid to late 80s to well into the 21st century. And so we did this by combining work in archives and oral history with the analysis of a large data set. And this data set was developed in collaboration with 3 colleagues at the University of Edinburgh that are experts in bibliometrics, social network analysis, also computer-assisted processing of big data. And it was formed by 2 collections. We firstly compiled all the submissions of yeast, human, and pig DNA sequences to the European Nucleotide Archive. As probably you know, it's a global repository that is curated by the European Bioinformatics Institute, the British Edinburgh, as Sabina was putting it. And but it is the entries of the European Nucleotide Archives are synchronized with the 2 other global repositories in Japan and in the US, the GenBank database. So one can retrieve, at least theoretically, the same information regardless what database they query. And also from the late 80s onwards, researchers, and especially those outside of the corporate world, were increasingly compelled by their funders and by their institutions to submit the DNA sequences to this repository prior to the publication of the sequence. So we chose a different chronological window to collect this information for each species, and the window was set so it would encompass the genome project of each of the species, and it would also include some years before and some years after this genome projects. And by doing this, we managed to gather almost 13.5 million DNA sequence submissions. And then the other part, the other collection of the data set, was the first description of these sequences in the scientific literature. We used two standard like bibliometric databases, and with the same time window in each of the three species, we retrieved almost 30,000 articles. And this is because some of the articles reported various DNA sequence submissions, in some cases thousands of them. And also towards the end of the period, lots of the sequence submissions were published. That's why there are much less articles than DNA sequence submissions. So we gathered a team that included not only the quantitative social scientists and not only historians and philosophers of science like Jean and myself, but also, for example, social scientists with specialized knowledge in yeast genetics or pig genetics. Also, social scientists investigating more broadly the sociology of scientific collaboration or innovation ecosystems. And then for the DNA sequence submissions, we look at what were the main institutions that were submitting DNA sequences in different time periods. And then with the publications, we created co-authorship network visualization. So this work with the dataset was published in 2022 in a special journal issue. So it was published 1 year before the book. And in fact, one of the papers, this one, kind of addressed these competitions between Tiger, Celera, and the public and charitably funded Human Genome Project, and why, for example, the genome of Tiger and Celera Genomics was never considered a reference genome, and the other was. So it is true that that still needs to be worked a lot, the standards of quality, but that was a bit of a side project that was addressing one of the papers of this special issue. But it was published before of the book and constituted the foundation on which we build the narrative of the book. So the book is divided into 3 parts. And in the first part, we present how in the mid to late 80s and early 90s, genomics was a fairly diverse field that encompassed quite a variety of activity, heterogeneous institutions. And in order to solve this, we look at the international project to sequence the yeast genome and the first attempts at mapping and sequencing the human genome. Then in the second part, we address the emergence of the reference genome as a research object. And we do that by looking at 2 different reference genomes, the human reference genome and the pig reference genome. And this is because these reference genomes were determined almost one after the other chronologically, the human reference genome in the late 90s, the pig reference genome in the very early 21st century. But in spite of this, they were determined using very different methodologies in very different ways, and they kind of perform a very different function, the human and the pig reference genome. And we account for this diversity by coming with the term genomicists in the book. So genomicists are the members of the communities that are behind the production each reference reference genome, and because of that, they sort of co-construct the reference genome. So there is a bidirectional interaction between these communities of genomicists and the reference genomes. So the genomes will fulfill the necessities, the specific necessities of a particular set of genomicists, and because of that, the affordances and limitations of the human reference genome are different from the pig reference genome and different from the yeast reference genome. And then finally, in the third part, we saw how the availability of these reference genomes makes the diversity of genomics visible at rest. So there is the impression that this diversity has narrowed down because of the reference genome, but as Sabina said, this is a bit of an illusion, almost a historiographical artifact. And by looking at how the reference genomes are used in practices like annotation or are interlinked to other biological data, such as protein sequences, we can see again how genomics is a diverse field. So because of this structure, in the first part of the book, we compare the history of genomics with the we claim that the history of genomics, at least as a first glance, may resemble the Orglass model that historians such as Ana Barahona, Edna Suarez, or Hans-Jörg Reinberger have proposed to kind of challenge the historiography of research into heredity. And they have claimed that this Orglass model made us believe that in the late 19th century, research into heredity was diverse, that there was heredity looked as a property with that could adopt lots of different forms and could be attributed to a variety of factors. But then with the rediscovery of Mendel's laws around 1900 and up to the late 20th century, the perception of heredity narrowed down massively to be equated with transmission of genetic information from parent to offspring. And then in the late 20th century, with the reunification of genetics with developmental biology and evolutionary biology, there was again this impression of heredity being a multifarious and multifactorial phenomenon. And similarly, at a first glance, in the historiography of genomics, we may think that there was a time for the reference genomes in the 80s and early 90s in which, for example, practice such as genome mapping or the development of DNA libraries were as important, if not more, as sequencing genomes, and there was attention to variation within and across species as much as to the creation of a consensus reference sequence. But then between 1996 and 2003, especially because of the publicity that the Human Genome Project and the determination of the Human Genome Sequence attains, genomics starts to be equating much more narrowly to the sequencing of the full genome representative of a species, of the reference genome. But then again, with the availability of these reference genomes after 2003, it looks as if genomics have recovered its diversity. But by considering the genomicists, the communities of genomicists, as the sun that goes down through the hourglass, as the people, the communities, the agents that make the history of genomics, we can achieve a much more dynamic model, a historiographical model for genomics, and we can start challenging this rigid hourglass model. So, for example, if we examine the community of yeast genomicists via the co-authorship network that we derive from the sequence, DNA sequence submissions of yeast to the European Nucleotide Archive, we see a somehow fragmented community with a core formed by mainly European institutions that have collaborated very closely and very strongly in the description or the publication of yeast DNA sequences. And then we have a periphery, a much more dispersed periphery of mainly American and Japanese institutions. And in the book, we explain this contrast in the data visualization by identifying two different strategies to sequence the yeast genome in the late 80s and early 90s. One strategy that was sponsored by the European Commission, so the yeast genome has been mapped during the 1980s, especially at Washington University in St. Louis and the University of Berkeley in the United States. But in 1989, the European Commission sponsored a project to sequence the yeast genome from that map, and this European Commission funded project attained at the end more than 80 institutions in several European countries. And these institutions have been involved in many areas of yeast biochemistry, yeast genetics, and by sponsoring this sequencing project, the European Commission wanted to foster collaboration among their different member states. So we identify this strategy as the distributed approach to genomics, and we contrasted this distributed approach to genomics to a concentrated approach to genomics, mainly in the US, but also in Britain. So in the distributed approach to genomics, the European institutions sort of pooled their efforts, their techniques, their expertise, and they would little by little sequence the yeast genome and use that information in their own biochemical and genetics research. Whereas the concentrated approach involved the creation of a new type of institution, the Genome Center. There were mainly three at the yeast phase: the Sanger Institute, Washington University, so one of the institutions that have mapped the yeast genome, and the University of Stanford in the US. And this new type of scientific institutions, what they did was to produce large amount of sequence information by using industrial factory-inspired methods. And the production of this largest amount of yeast sequence information was not to use that information in yeast research, but for creating a technological platform to then sequence larger genomes, especially the human genome. So in the case of the community of human genomicists, we also saw a contrast in the co-authorship network visualization between the institutions that were more central in this network, so the institutions with more co-authorship ties with other institutions, and then the institutions that have submitted the largest amount of sequence information to the nucleotide archive. So there was not overlap between the institutions with lots of co-authorships and the institutions that had submitted a lot of DNA sequence. And in the book, we interpret this contrast again by identifying two different ways of doing human genomics. The most central institutions in the network were laboratories in the field of human and medical genetics that, as the institutions of the European Yeast Consortium, they have pooled their expertise, their techniques, and come together to map and sequence increasingly large areas of human chromosomes connected to diseases. For example, the Huntington disease, and there was a Huntington disease consortium. So they would create a consortium like the yeast genomicists, but unlike the yeast genomicists, they didn't want to sequence the whole human genome. They wanted only to map and sequence the parts of the genome that were relevant for the disease they were investigating. And this contrast again with the genome centers that in the 1990s were shifting from yeast to human, and that they wanted to map and sequence the whole human genome. And what is interesting is that during the early and mid-1990s, these human and medical genetics institutions had a very strong presence in the history of genomics. For example, most of the authors of the first issue of the journal Genomics that was published in 1997 were human and medical geneticists. And why? because these laboratories would want, apart from mapping and sequencing a part of the chromosome, also to publish a paper in which they would describe what was the significance of this data for the disease they were working on. Whereas in the case of the genome centers, the priority was to produce this data rapidly and comprehensively, and there was not such a priority to publish. That's why the genome centers are the central in the co-authorship network, and they are very central because they publish the sequences as well. Also, human and medical geneticists were behind the first national projects to map and sequence the human genome. That's another point we make in this chapter, that there was not just one genome project, there were several genome projects in different countries. But around the 1990s, the center of gravity of genomics starts shifting from the human and medical geneticists, that at least in the big whole genome projects become more and more marginal, and towards the genome center. And we devote a chapter in the book to explain this shift in the center of gravity of whole genome genomics. And we do this by looking at, Sabina was saying, at the history of the Sanger Institute, the Genome Center in the UK, founded between 1992 and 93, and that has been involved in the three species, in the genome projects of the three species we addressed, yeast, human, and pig. We kind of identified this Sanger Institute in the vision that the World Contrast had of genomics as a science that would determine the whole sequence of different genomes, especially the human genome, and would make this information available, open access to the research community. So this vision materialized in the Bermuda Conference of 1996. Another main player for this was the co-elucidator of the double helix of DNA, James Watson. And in this Bermuda Conference, a small number of scientists, most of them behind these large-scale genome centers, agreed to sequence the whole human genome and make this information available unrestricted, not for themselves, but for other researchers to conduct their own investigations. Among them, the medical and human genetics that they have displaced from the core human genome project. So this effort, and we put this meeting at the neck of our genomic surplus, because this meeting and the visibility it attained led to equate one particular strategy to map and sequence the human genome with a single and successful and coherent human genome project, whereas the other national initiatives that we have in the previous chapters were largely forgotten in the historiography of genomics, and it also led to equate this particular version among many of human genomics to the whole field of genomics. As Jim is going to discuss now, if we shift our historical lens from human to other organisms, for example, the pig genome, we can see that the story is very different, and the way the pig reference genome was produced and used, the function itself for its community of genomicists is very different. Now, to you, Jim.
So, the topic of chapter 5 is the sequencing of the pig reference genome, primarily involving the community genomicists involving existing pig geneticists, but also the Sanger Institute. And some of our, some of the sort of key aspects of our thinking about the reference genome and its potential role in the development of genomics and what genomics enables or maybe occludes for different species came from some of our network-based analyses and quantitative analyses of the data sets that Miguel has described. So this is an example of the PIG co-authorship, institutional co-authorship. I'm not going to go into detail about this. Obviously, if anyone's got any questions about it, I can go into more detail about some of the interesting patterns and relationships that inform that, as well as obviously some of the quantitative aspects. But the key thing I really wanted to point out is the way that it led us to think about the reference genome, or the advent of the reference genome, the data and infrastructures associated with it, as very much an inflection point in the kinds of work that become possible and the kinds of work that no longer be considered meaningful contributions, research contributions, or publishable research contributions. So we see with the advent of reference genomes and reference genome data for the various periods for the 3 species, did change the patterns of collaboration as a result of the advent of an availability of this form of data. So certain activities are no longer as significant. Others enabled by the availability of a reference genome as an object, as a standard, as a resource that can then be constituted as a scaffold for the further investigation of the genomics of a species or other aspects of the biological variation of a species. So it enables new forms of research, but the extent to which it's able to link to new forms of research and generate new kinds of data, link to them, is very much contingent on the way in which this particular object has been produced and who is involved, who constitutes the community of genomicists involved in creating it. And so reference genomes are standard consensus DNA sequences for usually a species. They're defined and stored in large-scale data infrastructures such as the National Centre for Biotechnology Information in the United States, their RefSeq database. You've got the Genome Reference Consortium, and also you've got the Ensemble browser and pipelines associated with the European Bioinformatics Institute, and often these are presented in genome browsers, which this is too zoomed out to show the A's, C's, and G's, but you can zoom in enough to see the actual sequence or bases. But zoomed out a little bit more, you can see other features such as genes annotated and arrayed along it, and different tracks that a user can select to identify different different objects that are annotated and aligned to the genome, investigating this way. And the key thing is, sort of picking up on what's been emphasised in the introduction, was that these are very much constantly more or less updated and revised, and not necessarily just updated and revised according to one single definition of quality or validation, but the ways in which a reference genome or any sort of genome sequence is assessed evolves over time. It depends on who constitutes the community of genomicists that essentially steward the reference genome for any given species. So there may be different epistemic goals that these communities have for the reference genome that they look after. So in terms of how they're constructed, as a sort of schematic for how this is done, we can first identify a source of DNA that one wants to sort of use and to construct a DNA library out of, to store it in yeast artificial chromosomes or bacterial artificial chromosomes. And this often involves the actual creation, discovery, or identification of a population in itself from which one can sort of draw the DNA and have that eventual reference sequence define that. And then one can construct maps of various kinds of the genome from the DNA libraries. Use some of those maps in the sequencing. Sometimes it's possible to sequence directly from the DNA libraries themselves, and modern genomic methods are not necessarily going to need all these stages. The sequencing of the sort of smallish stretches of DNA then need to be stitched together, a process called assembly in multiple stages often, and then in order for the genome to actually be useful, meaningful, and usable in any form, it needs to be annotated with all the various features such as genes and other genomic variants marked along them. So researchers can, essentially, the idea is they pick that up and then they can use it to either construct their own genomic resources related to other forms of variation and make use of it in some way. And the important thing to stress here is that this is not, although presented in a unilinear way, it's not necessarily, it doesn't necessarily operate in that kind of unidirectional way in reality, and it's a much more dynamic and recursive process than that, with what are conceptually later stages of the process, such as annotation, informing the way the earlier stages, conceptually earlier stages, are conducted. So, for example, the annotation of the genome using known data on other organisms or known data on, for example, the identity and location of particular genes in the genome can be used to evaluate the assembly, can be used to evaluate the pre-existing mapping, or even the source of the DNA and how partial unrepresentative that may be. If, for example, a known gene is missing from the assembly. And it can be inferred in various ways, whether this is a result of an error with the sequencing process, whether it could be assembly, or something else. And this occurred in a number of cases in the pig genome project, because there was such an entanglement between the existing community of animal geneticists who've been working on the pig, mapping the pig for decades, were able to identify particular assembly errors. And this not only led to improved sequencing and assembly of those particular regions, but also changes to the actual process conducted by the Sanger Institute and the EBI themselves, so actually change the pipelines as well as the actual immediate result. And there's this kind of recursive and two-way process will become relevant for considering the role of the reference genome more widely a bit later on. So in terms of looking at the reference genome genomics across the three species, so Miguel has already summarised the case for yeast and human, where you have this the community of genomicists for yeast involving this distributed model of sequencing, whereby yeast biologists themselves were involved in sequencing large regions of the genome, and then immediately able to interpret and use the data that they generated in their own research programs, which themselves were the motivation for sequencing those particular areas. In human, though, there was a separation of the medical and human geneticists who'd previously been leading mapping efforts and some sequencing efforts from the International Human Genome Sequencing Consortium, and there was the concentration into a smaller number of specialist genome centres and the development of specialist genomicists, so people who were not performing genomics for particular research purposes, but to actually further the ends of genomics itself almost. And this is where you get the development of certain these annotation and assembly pipelines, such as Ensemble at the European Bioinformatics Institute. The pig, on the other hand, the swine genome sequencing consortium that emerged in the early 2000s was comprised in large part by animal geneticists working towards generating data and knowledge and tools for helping to improve the efficacy of livestock breeding, as well as a certain body of immunogeneticists working in particularly France and the United States, who've been involved in mainly European-led, European Commission-funded projects to map the pig genome from the early 1990s onwards. But they, having previously been involved with the Sanger Institute in physical mapping, engaged the Sanger Institute as a contractor for the actual sequencing process. The Sanger Institute, by this point, had been involved in sequencing, I think, about a third of the human genome, and developed all these refined pipelines, as well as the kind of industrial model of sequencing itself. But the interesting thing is that although the contractor model just looks like the community of pig geneticists had just kind of given a big chunk of money to the Sanger Institute and let them get on with it, there was actually quite an ongoing interactive relationship with the Sanger Institute from some of the leading figures in the pig genetics community. And one manifestation of this was the fact that there wasn't enough money to sequence the whole genome as much as possible in as much detail and depth and quality as possible. So the community itself needed to identify particular areas of the genome where they wanted the Sanger Institute to focus in on sequencing of greater coverage and quality, because they knew those regions would be most pertinent to their own research and application of that research. There was also the community was involved in some aspects of the assembly, using their own knowledge of the genome of the pig to actually help assemble certain parts of the genome that had become problematic in the particular pipelines of the Sanger Institute, because these relied on to a large extent on how the quality of the data affected the quality of the sequencing data affected how good the assembly pipelines worked, and some of it was quite rough and ready, so it needed this additional input from the community itself. And then there was this very interesting model of annotation which developed in collaboration between the Sandwich Institute and this community that was labelled as distributed community annotation. So this is whereby, you know, there was a first pass of the annotation going through the pipelines of the Sandwich Institute itself, of the Havana group, Universal Annotation Analysis Group, but also this model of distributed community annotation, whereby the Sandwich Institute developed tools for researchers to go away and actually annotate regions of the genome of interest themselves, gave a certain group of them actual training in person at the Sandwich Institute to do this, and they could then go away, do the annotation of the regions of interest, train up other people in their labs or local areas, and then contribute this annotation data to back to the Havana group, where they would assess it, validate it, and incorporate it in the annotation of the eventual reference genome. So 3 different models of the way that reference genomics proceeded, 3 different communities of genomicists and the relationships to the existing forms of biological research concerning those animals, and 3 organisms, and 3 very different kinds of reference genome as a result. So I want to sort of briefly look at the kinds of genomics work that was enabled by the advent of the reference genome. And we prefer the term post-reference genomics to post-genomics, which we can go into if anyone's interested in that. But it's just to provide a more neutral term to basically say this is simply the genomics that occurs for a species after the reference genome is produced. And we broadly identified 2 modes of post-reference genomics. A systematic mode, which is essentially examining patterns of variation and diversity and the processes that generate them, and functional, which is essentially assessing the effects of genomic variation. And the relationship between these 2 modes differs across the 3 species. So, for example, systematic mode and functional mode, the ENCODE project of human genomics. So in human genomics, for example, there was very much successive, parallel, but largely separate functional and systematic projects that were pioneered and conducted largely by the same large-scale genomics centres and institutions that had arisen and dominated the eventual human genome project. And that these kind of modes only very latterly came together through this sort of large-scale infrastructure, large-scale data infrastructures much more recently. On the yeast side, however, the community of yeast biologists that had been involved in the actual yeast sequencing then came together again, and it's largely the same constellation of people to do functional genomics, the Eurofan, the European Functional Analysis Network. And then very soon after that, engaging in more systematic projects to assess the diversity, not just of Saccharomyces cerevisiae, but also related species as well. But they very quickly into that came to appreciate some of the synergies between these 2 modes of work and actually articulated it quite explicitly. And these 2 modes then became quite entangled from there on in. In the case of the pig, the entanglement between systematic and functional occurred far before the actual genome sequence project was underway. There was an appreciation, especially mediated by the concept of animal genetic resources. Bear in mind, these are people that are largely involved with interrogating the pig genome because they want to be of use to the livestock breeding industry. So they're trying to find genes, whether it's in a population of Mongolian pigs, that might be useful for introgression, for inserting essentially into commercial populations of pigs. And here the examination and the surveying of diversity goes hand in hand with the sort of more functional concerns of what these genes are going to do and what effects they're going to have on the pig's phenotype. So they're entangled from the very beginning, and that remains throughout reference genomics and post-reference genomics as well. So thinking about what reference genomes actually represent, they're a particular form of abstraction and possible variation manifested across a species. But for each of the 3 species, they're sort of abstracted in very different ways. So for yeast, there was a very definite decision about the use of a particular S28AC, a particular laboratory strain, quite distinct from wild populations. But it's a standard strain that's used in laboratories across the world. So they were aware of how different the genome of this strain is to wild strains. They later sequenced some of the wild strains and compared them, but they, they had a very clear idea of what this really represented. There was a far more arbitrary and ad hoc origins for the human reference sequence, and this is the International Human Genome Sequencing Consortium one, not the cellular one, where they placed an advertisement in an upstate newspaper in New York State for volunteers to just contribute DNA, and this constituted most of the DNA which was in the actual eventual human reference genome. There was no real conception that it needed to be a sort of sampling of diversity of humanity or anything like that. It was North Americans of European descent. They had certain reasons to to sort of underpin why this sort of arbitrary ad hoc approach was valid. But the interesting thing is despite it being quite sort of arbitrary and ad hoc in this way, they very much viewed the reference genome as representing Homo sapiens as a whole in an almost sort of not almost metaphysical way, but really in a metaphysical way. On the pig, on the other hand, it was very much, they knew the name of the pig that most of the DNA for the reference sequence came from. T.J. Tabasco, a North American Duroc sow, that's a North American domesticated breed, who they'd already extracted DNA to create DNA libraries for other forms of research at the University of Minnesota, and that they actually got the group that created the DNA libraries for the Human Genome Project to do that. So it was already around. There were quite extensive libraries. There were other resources associated with T.J. Tabasco, the cloned offspring, for example, and so they just used that. They used whatever was lying around, adapted it, reused it, and so there was a very instrumental conception of what the pig genome represented. It didn't represent the scrotum as a whole. They were aware of how distinct T.J. Tabasco was genetically from many other pigs around the world, and they had an instrumental conception of the genome as a whole. It was a rough and ready research tool for the immediate purposes they wanted to put it to. They could improve it in future. They could get a pot of money here, a pot of money there to improve this bit, to improve that bit, to make use of it in new ways. But they had a very instrumental conception of that object. Now, reference genomes have been criticised for not reflecting, particularly the human reference genomes, have been criticised for not reflecting the breadth of genomic variation across the species, for not necessarily including common variants, for including very rare ones, maybe pathogenic ones, and also for this partiality of deriving it from people of largely European descent. So there are all sorts of potential issues there with the use and application of that genome on non-European populations or non-European ancestry populations. And there's one criticism that I think we thought was particularly striking. The data assembly that made up the human reference sequence reflects a highly specific process operating on highly specific samples. And they describe it as a type specimen. So a type specimen is an object in taxonomy. It is the name-bearing type, so it is an actual specimen collected from nature that then becomes essentially the anchor for a given species name, to which all additional samples collected in nature can be compared to it and see if it's, broadly speaking, a member of the same species, same morphology and so forth. But there's no consideration necessarily of whether the actual type specimen it represents the most common morph of a particular species in nature, whether it's completely idiosyncratic or whatever. So on that basis, you get these samples from these people in Buffalo, upstate New York. How do you know that they are representative of the human species as a whole? The type specimens aren't completely fixed across all time, but they are largely fixed. And so there's this kind of criticism of arbitrariness and fixedness of reference genomes, particularly the human reference genome. This is what these criticisms lay at. But we think that's kind of mistaken to think in those terms, or at least it kind of misses the point that the reference genome isn't necessarily meant to represent something, but the value of a reference genome can be accessed to according to the infrastructural role it plays in enabling new genomic resources, enabling other forms of data to be created and to be related to each other. And in this respect, we think that to some degree they're analogous to model organisms, which allow large-scale comparative work across species, integrating a range of disciplinary research approaches. But not to the extent that a model organism is representative of a particular phylum or representative of a particular biological process or mechanism, but the extent to which it can actually enable and seed other forms of research beyond itself, as well as within itself. And so I think rather than asking, what do reference genomes represent the species appropriately, we should better ask the question of how reference genomes enable the seeding of webs of linked reference resources, characterizing different aspects of the variation of species, and foster new forms of biological research and enable application of research. So just to sort of detail the idea about webs of reference, this is a sort of schematic of types of particular reference resources that might be linked to a reference genome or generated using a reference genome. So we might have, for example, a reference sequence of a particular population, breed or strain, a reference sequence in part, sequence in part or in whole of an individual, a reference sequence of a related or maybe not that well-related species. That can be constructed through scaffolding on and validated using the existing reference genome. We can have the use of the reference genome as a comparator to apprehend variation for just identifying variants, building tools such as SNP chips or microarrays that can then be used to genotype a large number of individuals and then greater appreciate the extent and the range and structure of variation of different kinds across a given species or population. And just as with the construction of a reference genome not necessarily being a unidirectional process, nor is the role of a reference genome in seeding this web of linked reference resources, that as these reference resources are constructed in part using the reference genome, they can allow researchers and the community as a whole to gain a greater appreciation of the range and extent and prevalence of variation, particular variants in a populational species, and therefore inform and enrich the updates and revisions of the reference genome, either by identifying errors maybe, but also by identifying what may be common variants, less common variants. Now that's not necessarily that the most common variants are definitely going to be built into new versions of the reference genome. It may well be that different communities have different aims, epistemic goals for what the reference genome is supposed to do for their own particular research and application aims. But the greater appreciation of variation enabled by the reference genome at least allows these processes to occur, and a particular community of geneticists to amend the reference genome to improve it according to their own goals and standards. So just as sort of interim conclusions for us to give some indications of future research, so reference genomes are considerably more revisable in dynamic relationships with related data and resources than type specimens are. Type specimens aren't fixed, but reference genomes are far more dynamic objects. But how that changes depends on the community of geneticists and its composition that is stewarding the reference genome. The development of a web of reference, its increasing connectivity and exploration of variation of space constitutes quite a distinct form of epistemic iteration. So epistemic iteration being the development of successive stages of knowledge and concerning a particular object or theme, towards the advancement of a particular epistemic goal. So, for example, in the physical sciences characterized by Hasok-Chan, the development of standards is far more sort of an epistemic iteration that constitutes a convergence upon fixed points. The generation of standards and other kinds of resources in genomics, on the other hand, is very much a radiation from multiple nodes that get created to explore this sort of web of reference, to explore the variational space. So it's a much more sort of characterized by radiation rather than convergence. And the genomics age translation, to the degree that it affords the generation of rich, extensive and recursive webs of reference. And this relies on the history of the creation of the reference genome in the first place and the particular involvement of communities and the community genomicists that steward it. Now, I'm not going to go into too much detail about this as I sort of come to a close, but thinking about the ways in which different communities stewarding involved in stewarding particular reference resources can explore the web of reference in different ways, for example, for different species, led me with David Ingram, a collaborator, to come up with the concept of veripraxis to sort of capture the ways in which the differential sort of exploration of this kind of space of variation. So we define veripraxis in connection with a paper on DNA barcoding as a set of practices involving the apprehension, measurement, investigation, and analysis of different forms of variation. It both captures forms of variation identified and used in the articulation of them in particular domains. And what I want to do going forward is to sort of really develop this concept which is aimed to foreground the ways in which variation is something that is the exploration of variation is very much characteristic, distinctive, and constitutive of biological research. And the formulation of this concept is presented really to try to operationalize that, to find a framework for more or less systematically investigating how variation is used and how the use and interpretation and treatment of variation can change over time in particular research programs and endeavours. Okay, so there's sort of two large questions about that underpin the prompts how forms of variation could conceptualize, explain, and theorize in relation to each other, and also it prompts that the thinking about what is the situation of a particular variopraxis in the historical context, trajectory of research program, and so forth. And there are all sorts of sub-questions then about characterizing forms of variation and in a given variopraxis, what forms of variation are related and how they relate to other aspects of a research program the term prompts. And so to do this, to explore variopraxis and also the particular forms of epistemic iteration that operate in life sciences research, I want to map webs of reference capturing how this omnidimensional variation space is explored and how these connections change over time. And I think yeast is probably the best way to do this because we've got a lot of data on yeast already, but also the yeast community, biology community, has thought of increasingly sophisticated articulations of the relationship between representations of genomes that they develop themselves. So they've conceptualized Saccharomyces as a modern genus and very much think in those terms, and also articulated ways in which. The increasing functional understanding of the genome can inform the increasing systematic understanding and vice versa. And I want to take to investigate how communities apprehend and investigate variation, look at how, in more detail, about how genetic variation and diversity has been investigated in the pig genetics community, because there you've got the intersection of a sort of horizontal and comparative inferential form of epistemic iteration alongside the radiation that we've identified. But also a field of study, embryo phenomics. There's a research group at the University of Plymouth doing this, where they're increasing these scales and dimensions of phenomic data collection and analysis, using various automated video analysis and machine learning and so forth in developmental ecophysiological research on freshwater and marine snails, for example. So I'll conclude there. There's some references I can ping back to if anyone's interested. So thank you very much. I appreciate you hosting us today. We thank obviously everyone that's involved, helped us with this project. I want to mention two more names on the project team: Rodrigo, who helped in cleaning up the massive amounts of data, and Yamo, who helped proofread the book. And obviously thank you to the European Horizon 2020 program as well. And there's a link to the book that's a bit easier there if anyone wants to use that.